Hello and welcome, everybody. Welcome to the Drug Hunter Flash Talks today. I'm your host, Dennis Koester, Director of Industry Research and Relations here at Drug Hunter. And it's my pleasure to host our very own Lew Pennington today. Good morning, Lew. How's it going?
Good morning. It's going well.
So, Lew joined Drug Hunter here as a scientific director in 2024. And prior to that, he served as the head of platform discovery at Chimera Therapeutics. And at Chimera, he co-invented the first molecular glue degrader in the portfolio. And this discovery resulted in the launch of an immunology drug discovery program and the initiative to build a molecular glue degrader franchise. Prior to that, he was at Alchemius and he was a research fellow. And there he initiated the orexin 2 receptor agonist program and served as chemistry team leader. And that resulted in the invention of a clinical drug and the initiative to build an orexin 2 receptor agonist franchise. Over the course of his 30 years in medicinal chemistry, Lew has contributed to numerous drug discovery projects on all stages, and that resulted in 2 clinical drugs, 4 clinical candidates, and Lew has also authored and co-authored 33 peer-reviewed publications, 18 granted US patents, and 34 World Office patent applications. And in addition to all of that, as if that wasn't enough, Lew also basically had a lot of research interests and mainly defining concepts, strategies, and tactics for multi-parameter optimizations in modern small molecule drug discovery. And that includes structure-brain exposure relationships, non-covalent sulfur interactions, the necessary nitrogen atom that we'll hear more about today, positional analog scanning, including the nitrogen scanning, and holistic drug design as well. And notably, the necessary nitrogen atom and the nitrogen scanning concept helped inspire the creation of an area of synthetic chemistry that is known as skeletal editing. Lew previously served a variety of roles as synthetic and medicinal chemist at Amgen, Array Biopharma, and Eli Lilly, and he earned his bachelor's at the University of Michigan and his Ph.D. under the mentorship of Larry Overman at the University of California in Irvine. Great pleasure having you today, Lew.
Yeah, thank you, Dennis, for that really kind introduction. It's great to be here.
Yeah, awesome. And as usual, as excuse me, as it's tradition in the Drug Hunter Flash Talks, we also want to ask you a few warm-up questions. So maybe can you let us know a little bit more about what inspired you to start a career in drug discovery?
Yeah, it was really a series of educational, professional, and personal experiences over my lifetime. So, I went to a rural school in Michigan, but I had a spectacular chemistry teacher, Charles Pierce, who really made me love chemistry. So that inspired me to go to the University of Michigan and major in chemistry. And when I was there, they had a spectacular program where the freshmen could take organic chemistry rather than waiting until their sophomore year. And that's where I decided, oh, I got to be an organic chemist, a synthetic organic chemist. But as I was getting ready to finish my bachelor's degree at Michigan, I didn't really know if I wanted to go to graduate school or even sort of what area of chemistry I wanted to pursue or in terms of the industry. And so somebody said, well, get a job. And I said, well, where should I get a job? And they said, well, why don't you go to a pharmaceutical company? Because they make the coolest molecules. So I was like, okay. So I went to Eli Lilly and I was in their endocrine research group. I was a medicinal chemist there. And that's where I learned that I love medicinal chemistry. I love drug discovery. And I had a great supervisor there, Tim Gracie, who managed to get me into Larry Overman's group because I wanted to get that PhD. And Larry was just a wonderful person to work for, as folks know. And once I was getting ready to finish up my PhD, I decided, okay, let's go into the biotech industry. I didn't really want to go back into big pharma. So I've sort of moved around in the biotech industry since then. And, like a lot of people, I have people close to me that have gotten sick. And it's really wonderful to be able to harness my love for chemistry to try to help people. And so, I cannot think of a nobler purpose than that.
Absolutely, yeah, very, very inspiring. And I think a lot of people trace their career beginnings back to an early mentor, which is very, very important. Emphasizes the importance of education. Yeah, with that, what's your favorite molecule today, Lou, and why?
Oh, well, it will probably come as no surprise to folks. ELK's 2680. This is the orexin 2 receptor agonist that I co-invented at Alkermes. It's currently in phase 2 clinical trials. So I'm excited about this, not just because I was, deeply involved in the invention of this molecule, but I believe that the orexin 2 receptor is going to do for CNS diseases what GLP-1 receptor has done for metabolic diseases. I think people don't yet fully understand just how powerful and broadly useful the orexin system is for CNS diseases. A lot of data is emerging. So keep an eye on it because this is going to be an incredible field, and a lot of companies are involved in this area. So lots of interesting work going on right now.
That sounds super exciting. Yeah, we're definitely monitoring the field, and yeah, there's already a lot of interesting stuff out there, but hopefully a lot more interesting stuff to come. Yeah, very exciting. Um, what are you most excited about in drug discovery today?
Oh, there's so many things. Like,
Hmm.
I guess just in general, what has me excited is how much incredible, innovative, small molecule drug discovery is going on. So, we've seen the emergence of a number of new therapeutic modalities over the last 30 years or so, and even more therapeutic modalities in the last 10, 15 years. But small molecules still constitute the majority of new drug approvals. And, I remember 15, 20 years ago, people thought small molecules were dead, that they were going to be displaced by all these other new therapeutic modalities. And here we are today in what I believe to be the most exciting time period for small molecule drug discovery ever. And so they're never going to go away. It's always going to be part of the pharmacopoeia. And I'm just excited and happy to be a small molecule guy working in this golden era of small molecule drug discovery.
I couldn't agree more, Lou. That's like you basically nailed it there. Yeah, I'm also very excited and have very similar motivations. Even we're seeing hearts in the audience here. So yeah, can you tell us a little bit more? I mean, you had a great career in medicinal chemistry, and you still have a phenomenal career and have a massive impact on drug discovery today. Can you tell us maybe a little bit more about the advice you have for the younger self, for your younger self, or for folks in the audience maybe that are starting their careers?
Yeah, my advice would be really understand that you are the captain of your career. And I didn't understand that when I was younger. I thought, go get an education, get a job, you'll be recognized for all of your contributions, and the opportunities will just open up to you. And that's really just not the case. Maybe for some people that happens, but you need to be very deliberate, in my opinion, about your career. And so, as early as you can, define your professional purpose, define your professional values, and define your professional goals. Now, these are going to change over the course of your career, but every few years or so, you can define all of those things, and then you come up with the strategies and the tactics to achieve those professional goals and live by your professional values. And hopefully, if you're lucky, you eventually realize your professional purpose. So, you know, one thing that I'm still hoping for is that I will be somebody who has made a key contribution to a marketed drug that's out there in the world helping people. And that's a core part of my purpose, and one of my core goals is a medicinal chemist in drug discovery.
Absolutely. Yeah. Thank you, Lou, for that advice. That's very, very useful advice for young folks. Thank you. Maybe as a last question before we get started, what do you most like about Drug Hunter now, as an insider?
So I love the mission of Drug Hunter. We are truly trying to transform the way that drug discovery knowledge is shared so that we can accelerate the development of medicines. Drug Hunter occupies a very unique space in the knowledge sharing universe. And I think, the sky's the limit for Drug Hunter. And I love what we're doing. It gets me out of bed every morning. This is another one of my professional purposes is to advance human health through sharing knowledge. And we're doing that in spades, I would say, at Drug Hunter.
Awesome. Yeah, thank you, Lou. And yeah, before you get started with your presentation, let me briefly share one or two features here on Drug Hunter that I wanted to kind of share with the audience today. One article that I want to share and that I want to kind of point out today is our review of the 2024 medicinal chemistry reviews here. And that's really in the spirit of knowledge sharing. So we partnered here with the ACS, and Antran helped write this article here. And really provides a very nice summary of a few drug discovery projects in major disease areas. Lou already mentioned here the CNS drug discovery area where orexin 2 receptor agonists are really exciting. There are a few other targets that are worth to be monitored here. There's also a nice introductory prologue by Tom Bale. And basically, he gives a lot of career advice. So if you're looking for some advice and reflections beyond what Lou just shared with us, that is also a great chapter to go to. Beyond that, you can find, really things about cardiovascular diseases, things about new targets and immunology and inflammatory diseases. Few very nice reviews here in the oncology field with, the super nice developments that have been happening in the PI3K alpha space, which we also have a lot of articles on Drug Hunter on if you want to dive a little deeper. New developments here in infectious diseases. There's topics in biology here as well, topics in drug discovery, macrocycles, and here the case histories of new chemical entities. And here I want to point out one thing that Lou said actually. Here there's a chapter about all the compounds that entered phase 3 clinical trials in 2023. And out of those compounds, the overwhelming number, 92% of those compounds are administered orally. And, they span a molecular weight from about 270 to 1,500. So that really, I think, highlights the what Lou has just said, that we're in like a golden era of small molecule drug discovery, and that they're really challenging targets that we're now trying to get a handle on. And I think with Drug Hunter, you will find our mission is to share knowledge for you to accelerate your drug discovery projects. Well, and I leave it with that for now. I don't want to take too much time away from Lou's presentation, and I'll hand it over to Lou to share with us more about the necessary nitrogen atom and how it can be used in property-based drug design.
All right, can you see my first slide, Dennis?
Yes, looks perfect. Thank you, Lou.
Great, just let me move these videos so I can see everything. All right, so firstly, I'd like to thank Dennis for giving me the opportunity to remind everybody how far we have come in small molecule drug design over the past several decades, and how far we have come in property-based drug design in particular. So I hope to show today how the necessary nitrogen atom can be leveraged for holistic property-based drug design. So why are we here? We're here because as drug hunters, we are engaged in one of the most challenging of human endeavors, the invention of safe and effective medicines with good tolerability and convenience. And these endeavors are challenging regardless of whether we're working on a potential first-in-class or a potential best-in-class medicine. Probably the greatest challenge is the selection of the right therapeutic target for a disease indication and validating that therapeutic target for that disease indication. Another challenge is the identification of hits that bind to that therapeutic target and validating that they do so. And then finally, another challenge is drug multi-parameter optimization, including optimization of pharmacokinetics, which is really about optimizing the drug concentration at the therapeutic target for the right amount of time. The optimization of pharmacodynamics, which is about optimizing the drug modulation of the therapeutic target in an appropriate way for a particular disease indication. And then finally, optimization of safety, the safety of the drug both off and on the target. So really what we're trying to achieve here is optimal drug target synergy for a particular disease indication. And this is why drug design is absolutely central to the discovery of a new medicine. As the drug molecule embodies the therapeutic hypothesis of a drug discovery program and encodes the properties, pharmacology, and safety of the medicine. And it's largely the responsibility of the medicinal chemist to design and synthesize the molecules that are tested as potential medicines, and to do so by integrating existing knowledge and the insights generated during a drug discovery program into a molecule that achieves the target product profile of the medicine. But of course, as we all know, drug design has evolved considerably over the last several decades. In the 20th century, we saw the emergence of rational drug design in the 1950s. And I think most folks listening today understand that a Nobel Prize in physiology or medicine was awarded for the development of rational drug design in 1988. Another interesting fact about that Nobel Prize is that's the only Nobel Prize ever awarded to a true medicinal chemist, despite the fact that medicinal chemistry has existed as a distinct discipline for over 100 years. After the development of rational drug design, we saw the development of quantitative structure-activity relationships in the 1960s, and then the development of ligand-based drug design in the 70s, computer-aided drug design in the 1980s, and structure-based drug design in the 1990s. In the 21st century, we saw the emergence of property-based drug design in the 2000s after the publication of the Rule of Five by Chris Lipinski and coworkers in 1997. And then in the 2010s, we saw the formalization of hypothesis-driven drug design. And then after that, we saw the development of artificial intelligence and machine learning approaches to drug design in the post-2010 era. So with all of these advancements and developments in drug design, modern drug design is not about doing drug design this way or that way, one thing or the other, but really taking a holistic approach to drug design where a medicinal chemist in a strategic and integrated sense uses all of these approaches to drug design. And with all of these developments in drug design and the evolution of drug design, oral small molecule drugs have also undergone a tremendous evolution over the past several decades. For example, if you look at a molecule like ibuprofen that was approved in the 1970s, and you compare ibuprofen to a molecule like lenacapavir that was approved in the 2020s, you see an incredible difference in the structural novelty and molecular complexity between these two molecules. Now, we believe this evolution of molecular novelty and complexity has been driven by a number of factors, a number of advances in drug discovery related science and technology. But we also believe that property-based drug design has played a key role in this evolution of oral small molecule drugs following the publication of Lipinski's Rule of Five in 1997. So just like drug design in general has evolved, property-based drug design has also evolved tremendously over the last several decades. So just to level set everybody, what is property-based drug design? Property-based drug design is the modulation of molecular and physical chemical properties to optimize pharmaceutical, pharmacokinetic, ADME, and safety properties. And in the modern era, there are a number of approaches to property-based drug design. So the first set of rules was the Rule of Five, like I mentioned earlier, that was published in 1997 by Chris Lipinski and coworkers. And after the publication of the Rule of Five in 1997, there was a tremendous amount of effort around the world at pharmaceutical companies doing property activity relationships to try to further understand how molecular and physical chemical properties impact pharmacological and safety properties. And over time, new approaches to property-based drug design emerged, such as composite metrics like lipophilic efficiency that emerged in 2007, and new individual properties like fraction sp3 that emerged in 2009. And that was followed by the emergence of algorithm-based approaches to property-based drug design, such as multi-parameter optimization scores, the first example of which was the CNS multi-parameter optimization score that was published in 2010. And then in the post-2010 era, we've seen a number of artificial intelligence and machine learning models developed for property-based drug design. So again, just like drug design in general, for property-based drug design, a holistic approach is also required. Again, it's not about doing property-based drug design one way or another, this way or that, but really taking a holistic approach. So what do we mean by that? So holistic property-based drug design is the strategic and integrated use of multiple property-based drug design approaches. So we believe property-based drug design is best viewed as chemical series specific, relative, and probabilistic. And what do I mean by that? What I mean is that for a given chemical series on a drug discovery program, a relative change in a molecular or physical chemical property has some probability of improvement in a pharmacological or safety property. We believe that individual properties can be used to focus design hypotheses and design proposals. Property activity relationships can be used to inspire, validate, or invalidate design hypotheses. Sets of rules can be harnessed as guidelines to assess the property space risk of design proposals. And composite metrics, MPO scores, in artificial intelligence and machine learning models can be used to further assess the risk and quality of design proposals. All that said, it is absolutely imperative that one adapts their property-based drug design strategy based on structure-activity relationship data using measured data. So now I'm going to pivot and talk about the necessary nitrogen atom, and I'll be weaving this concept of holistic drug design and the necessary nitrogen atom as I proceed with my talk. So what is the necessary nitrogen atom? Simply put, it's the exchange of an aromatic CH group with a nitrogen atom that results in at least 10-fold improvement in any pharmacological or safety properties. So for many years now, I've been preaching that this classical bioisosteer has very broad utility in this modern context of multi-parameter optimization and property-based drug design. And in situations where we don't know where to put that nitrogen atom, nitrogen scanning can be used to find the preferred positional analogs. So in the example I show here, this stems from the orexin 2 receptor agonist program that I started at Alkermes. And so at a certain point in this program, we desired to improve the metabolic stability of this advanced lead compound, compound 1. And we were looking at the metabolic stability as measured in human hepatocytes. So the hypothesis we had on this program was that if we could decrease the lipophilicity of advanced lead 1, we might be able to decrease the hepatocyte intrinsic clearance. So that then led to the question of, well, where do we put that nitrogen atom? There are 4 aromatic CH groups on this phenyl ring over here. Where do you put that nitrogen atom to get a beneficial effect on metabolic stability without having a detrimental effect on other pharmacological or safety properties? And we didn't know. So what we had to do is what medicinal chemists often have to do. We had to make and test all 4 of these positional analogs. So as you can see here, in going from this advanced lead compound 1 to its AZA analogs 2, 3, 4, and 5, we eventually got to compounds like 4 and 5 that display nearly 10-fold improvement in hepatocyte intrinsic clearance. However, as is often the case with multi-parameter optimization efforts, this improvement in metabolic stability came with some baggage. And in the case of compounds 4 and 5, that baggage was dramatically reduced potency. Additionally, if you compare the permeability and efflux profiles of the lead compound 1 with its AZA analogs, you see there's generally also some negative impacts on permeability and efflux. And this probably isn't too surprising given the increased polarity of the AZA analogs. Now, one can also imagine that doing this nitrogen scanning effort in the context of these complex macrocyclic compounds was quite challenging and required a tremendous amount of synthetic chemistry investment. And that indeed was the case. And that's why we're really excited about the emergence of this new area of synthetic chemistry called skeletal editing. And we're excited about skeletal editing because it allows for the late stage functionalization of advanced lead compounds and avoids the need for de novo syntheses of each positional analog, as was required in the previous slide. It also enables predictable, site-specific ipso-aromatic CH group to nitrogen atom exchange. So in the example I show here, this comes from the original science paper by Levin and co-workers that was published late last year. And this represents a partial nitrogen scan. So this starting material here on the left has 4 aromatic CH groups that could be replaced with nitrogen atoms. And in this case, they replaced 2 of the 4. So this represents a partial nitrogen scan rather than a full nitrogen scan. So in the first step of this process, the unselective borylation azidation sequence led to a mixture of these 2 azide analogs that were separable. And these azides were then advanced separately into the second stage of the process, the site-specific nitrene internalization, to give the 2 aza analogs shown here on the right-hand side of the figure. So this is a really exciting result. And I'm, very, very closely watching the development of this field because this has the opportunity to greatly increase the efficiency of our lead optimization efforts in medicinal chemistry and small molecule drug discovery. So one thing to keep in mind with this structure modification is that even though it's a very small structural change, it has a very big impact on molecular and physical chemical properties. For example, in going from benzene to pyridine, one goes from a resonance-stabilized aromatic ring system with an even distribution of electron density around the ring to a resonance-stabilized heteroaromatic ring system with variable electron density around the ring in a polar nitrogen atom bearing an unshared pair of electrons coplanar to the ring. Additionally, going from benzene to pyridine, one goes from a pi electron-rich system to a pi electron deficient system. So this structure modification has a number of effects on other molecular and physical chemical properties of relevance to drug design. With respect to resonance stabilization energy, molecular weight, and even the van der Waals radius at the point of substitution, this structure modification has a very small effect. However, because of the differences in electronegativity between the carbon atom of the CH group and the nitrogen atom of the pyridine, as well as the unshared pair of electrons on the pyridine atom, there's a very profound effect on polar surface area. You go from a molecule with no polar surface area to a molecule with 13 square angstroms of polar surface area. This structure modification also has dramatic impacts on basicity, hydrogen bond acceptor strength, dipole moment, and lipophilicity. So in addition to all of these effects on molecular and physical chemical properties, this structure modification also impacts a number of intra and intermolecular interactions, including electrostatic interactions, van der Waals interactions, hydrophobic interactions, and orbital interactions. And when one is contemplating the effects of this structure modification on these interactions, one needs to think not just about the CH group that was replaced and the nitrogen atom that replaced it, but also the ring system and the substituents on that ring system. And indeed, the ring system and its substituents impact molecular and physical chemical properties. So one thing to keep in mind with this structure modification is that the number and the position of the nitrogen atoms is critical. For example, in going from pyridine to its diaza analogs pyridazine, pyrimidine, and pyrazine, one sees a steady decline in basicity across this homologous series. With respect to hydrogen bond acceptor strength and going from pyridine to pyridazine, there's a very small change in hydrogen bond accepting strength. But both pyrimidine and pyrazine have much lower hydrogen bond accepting strengths than pyridine. With respect to dipole moment and going from pyridine to pyridazine, we see a very large increase in dipole moment. However, pyrimidine has a very similar dipole moment to pyridine, and pyrazine has no dipole moment due to its symmetrical nature. With respect to lipophilicity, this probably isn't too surprising. In going from pyridine to its diaza analogs, we see a fairly dramatic decrease in lipophilicity. And then finally, with pi electron density of these ring systems, in going from pyridine to its diaza analogs, we go from a pi electron deficient system to even more pi electron deficient systems. And this can have a number of impacts on pharmacological and safety properties of relevance to drug design. So in addition to all of these improvements in all of these molecular and physical chemical properties of relevance to drug design, the structure modification also can be leveraged for holistic property-based drug design. So this structure modification aligns very nicely with the rule of 5. In making a CH group to nitrogen atom exchange, there's only an increase of 1 Dalton in molecular weight. 1 decreases the log P by about 1 unit with the structure modification, and 1 introduces only a single additional hydrogen bond acceptor with the structure modification. And then, of course, there is no new hydrogen bond donor that is introduced with this structure modification. This structure modification also increases lipophilic efficiency, and it can do that either by increasing potency and decreasing C log P, or simply by decreasing C log P. This structure modification also increases the fraction sp3. So if there's a single sp3 hybridized carbon atom in the molecule, this structure modification decreases the total carbon atom count and increases the fraction sp3. So now I'd like to pivot and talk about some general trends that we and others have observed for this structure modification. With respect to generic effects on pharmaceutical, pharmacokinetic, ADME, and safety properties, this structure modification has a positive impact on lipophilicity, solubility, protein binding, and microsomal stability. It has a positive or negative impact on cytochrome P450 inhibition. So if you look over here at this table on the right, you can see that in going from the phenyl progenitor to the 2-pyridyl, 3-pyridyl, or 4-pyridyl AZA analogs, you can see that the 2-pyridyl analog actually has a lower propensity for CYP inhibition, but the 3-pyridyl and 4-pyridyl start to have a bit more propensity for CYP inhibition. And this is likely due to the more exposed nitrogen atom in the 3-pyridyl and 4-pyridyl analogs being able to bind to the iron atom of the heme in the cytochrome P450 enzyme. This structure modification generally has a negative impact on permeability, likely because of the increased polarity of the AZA analogs, but this isn't always the case. And I'll actually show 2 examples in the case studies where there is an improvement in permeability and efflux observed upon this structure modification. And although it's not shown in this slide, this structure modification can also have a positive impact on PXR activation, hERG inhibition, and Ames mutagenicity. And if you want to see examples of that, you can look at the references here, as well as the references that are cited in some of these articles at the bottom of the slide. So one final point I want to reemphasize, I made it on the previous slide and I want to say it again, the number and position of nitrogen atoms is absolutely critical. So when you're contemplating leveraging the structure modification in your lead generation or lead optimization efforts, I would advise you pretty much try all monoaza, diaza, and sometimes even triaza analogs possible, because you're going to be very surprised with some of the results you get, because a lot of times, like I said previously, you cannot predict a lot of these PK, ADME, and safety properties. So this structure modification has broad utility in drug design. So in a recent analysis of nitrogen heterocycles in approved drugs, they found that nitrogen heterocycles show an increasing occurrence in approved drugs. And in fact, from January of 2013 to December of 2023, it was found that 82% of drugs contain at least 1 nitrogen heterocycle. And this represented an increase of 59% of drugs from the June 1938 to the December 2012 time period. Additionally, it was found in this analysis that nitrogen atom containing heteroaromatic ring systems comprise a sizable and increasing proportion of nitrogen heterocycles in approved drugs. And in fact, they found that the pyridine ring system jumped ahead of piperidine as the most common nitrogen heterocycle in approved drugs in the 2013 to 2023 time period. So these trends are likely because medicinal chemists have come to prefer neutral, low lipophilicity drugs for their generally better behaved pharmacological and safety properties. So before I get into the case studies, I would like to make a fair balanced disclosure, just like is required when you're trying to market an approved drug. So I would like to emphasize there is such a thing as the superlative CH group or unnecessary nitrogen atom. And that means that pharmacological properties may be worsened by aromatic CH group to nitrogen atom exchange. However, I would like to say that in general, the physical chemical properties of the CH progenitors are generally less desirable than they are for the nitrogen analogs. Additionally, there are some common undesired effects that may be observed with the nitrogen analogs, including aldehyde oxidase metabolism and cytochrome P450 inhibition. Now, medicinal chemists have come up with a variety of strategies and tactics to get around these issues when they arise, and you can see a number of those examples in these references at the bottom of the slide, or even the references cited in those references. And then finally, I would like to say that the prediction of the effects of this structure modification can be challenging, particularly in a multi-parameter optimization context. So by all means, we should use predictive modeling when it's available and reliable, but that's often not the case. A lot of times we have reliable predictive models for one or two or even three of the parameters against which we're optimizing our molecules, but not all of them. And in these situations, it's absolutely appropriate to seek serendipity. And one strategic and systematic way of going about doing that is through nitrogen scanning. So for my first case study of the day, this stems from a program seeking positive allosteric modulators of the muscarinic acetylcholine receptor, M4. So in this program, they encountered a chemical stability issue in aqueous buffer at pH 1. And what the hypothesis on this program was that perhaps if they could decrease the protonation tendency of this azetidine nitrogen atom, they might be able to improve the chemical stability. And what they found was that by replacing this CH group with a nitrogen atom, they were able to afford a greater than 480-fold increase in chemical stability in aqueous buffer at pH 1. The structure modification also had a positive impact on human liver microsomal intrinsic clearance, but a negative impact on efflux. So to get around that issue, they found that if they replaced this hydrogen atom with a trifluoromethyl group, they could further increase potency, decrease efflux, and this led to compound 8, emraclidine, an M4 positive allosteric modulator. And I would like to note that emraclidine was a 2022 Drug Hunter Molecule of the Year runner-up. In the next case study, this stemmed from a program looking for inhibitors of poly-ADP ribose polymerase 1. They ran into an aqueous solubility challenge as measured in aqueous buffer at pH 7.4. So the hypothesis on this program was that if they could decrease the lipophilicity of this advanced lead compound, compound 9, they might be able to improve aqueous solubility. And what they found was that by replacing this CH group with a nitrogen atom, they were able to elicit a greater than 36-fold improvement in aqueous solubility. The structure modification also had a positive impact on potency, hERG inhibition, and efflux, but it had a negative impact on human liver microsomal intrinsic clearance. So to get to the clinical compound, they did another CH group to nitrogen atom exchange, in this case, exchanging this CH group with a nitrogen atom. And that had the effect of further increasing aqueous solubility, decreasing human liver microsomal intrinsic clearance, and increasing that hERG IC50 and really dialing out that hERG activity. And this led to compound 11, saruparib. This is a PARP1 inhibitor in the clinic. In a program looking for allosteric inhibitors of tyrosine kinase 2, they ran into a cellular permeability challenge as measured in CACO2 cells. The hypothesis on this program was if they could mask a hydrogen bond donor with an intramolecular hydrogen bond, they might be able to increase permeability. And what they found was by exchanging this CH group with a nitrogen atom, they observed a greater than 4.7-fold increase in permeability. The structure modification also had a positive impact on Cmax in vivo pharmacokinetic studies. So this is a nice example of adding a polar nitrogen atom and actually observing an increase in cellular permeability. So as folks know, deucravacitinib was approved a couple years ago, and this was actually a 2022 Drug Hunter Molecule of the Year. In a program looking for inhibitors of renin, they ran into a metabolic stability issue as measured in human liver microsomes. The hypothesis on this program was that if they could decrease the electron density of this indole ring system, they might be able to increase metabolic stability. And what they found was that by replacing this CH group with a nitrogen atom, they could engender an 8.2-fold improvement in human liver microsomal intrinsic clearance. The structure modification also had a positive impact on CYP3A4 time-dependent inhibition, but a negative impact on potency. So to get to the clinical compound, they had to do 2 more hydrogen atom to methyl group exchanges to basically improve potency. And then another CH group to nitrogen atom exchange, largely to further decrease the CYP3A4 time-dependent inhibition, and that led to compound 16, cytokinin, a clinical stage renin inhibitor. In a program looking for antagonists of the gonadotropin-releasing hormone receptor, they ran into an oral bioavailability challenge as measured in Wistar rat. So the hypothesis on this program was they could decrease the lipophilicity and electron density of this 2-chlorophenyl ring, which was identified as the primary site of metabolism in some preliminary metabolite ID studies, they might be able to further improve oral bioavailability. And what they found was that by exchanging this CH group with a nitrogen atom, they saw a 14-fold increase in oral bioavailability. And this structure modification also had a positive impact on hepatocyte stability. To get to the clinical compound, they exchanged this hydrogen atom with a trifluoromethyl group and this amide functionality with a sulfone. These structure modifications had the effect of further increasing oral bioavailability and decreasing hepatocyte clearance. And this led to compound 19, Bay1214784, a GnRH receptor antagonist in the clinic. And this compound was a 2020 Drug Hunter Molecule of the Year runner-up. In a program seeking inhibitors of cholesterol ester transfer protein, they ran into a CYP inhibition issue, including CYP3A4, 2D6, 2C9, and 2C8. So the hypothesis on this program was that if they could decrease the lipophilicity of this advanced lead compound, they might be able to decrease that CYP inhibition. And what they found was that by replacing this CH group with a nitrogen atom, they saw a 3.3-fold increase in the CYP IC50 for CYPs 3A4, 2D6, and 2C9. They had no effect on CYP 2C8. The structure modification also had a positive impact on potency, but a negative impact on NAV 1.5 and CAV 1.2 inhibition. So this is a nice explicit example of an unnecessary nitrogen atom, like I talked about earlier in the talk. And so to get around those issues, they found that if they replaced this nitrogen atom with a CH group, they could further increase potency, increase the CYP2C8 IC50, and dial out the NaV 1.5 and CaV 1.2 activity to lead to compound 22, MK8262, a CETP inhibitor in the clinic. In a program looking for inhibitors of DNA-dependent protein kinase catalytic subunit, they encountered a hERG inhibition challenge. So the hypothesis on this program was that if they could decrease the basicity of this imidazopyridine nitrogen atom, they might be able to dial out that hERG inhibition activity. And in fact, what they found was that replacing this CH group with a nitrogen atom, they saw a greater than 25-fold increase in hERG IC50. And this led to compound 24, AZD7648, a DNA PCS inhibitor that's in the clinic. The structure modification also had a positive impact on permeability and efflux. So this is another example of introducing a polar nitrogen atom and actually observing improved permeability and efflux. So for my final case study today, this stems from a program seeking inhibitors of phosphodiesterase 2A. In this program, they ran into a cardiovascular toxicity assay in a rat 15-day exploratory toxicology study. And the toxicity was only described as microscopic findings in the vasculature. They really didn't go beyond that. So the hypothesis on this program was that perhaps if they could further decrease the lipophilicity of this advanced lead compound, they might be able to dial out this cardiovascular toxicity. And what they found was that by replacing this CH group with a nitrogen atom, they were able to go from a compound that displayed rat CV toxicity to a compound that did not display CV toxicity. However, compound 26 still displayed cardiovascular toxicity in a dog exploratory toxicology assay. So to engineer out that activity, they exchanged this methylamino group with an azetidine group, which both increased the potency of the molecule and decreased that dog CV tox. And this led to compound 27, PF0518099, a PDE2A inhibitor that reached the clinic. And I forgot to mention that the initial CH group to nitrogen atom exchange also had a positive impact on human liver microsomal intrinsic clearance and a negative impact on potency. So to conclude the talk, hopefully I've shown you or gotten you to believe that an aromatic CH group to nitrogen atom exchange is a privileged structure modification for property-based drug design that can lead to the identification of necessary nitrogen atoms where a greater than or equal to 10-fold improvement in a pharmacological or safety property is observed. I would like to mention that two AZA analogs seem to have an increased probability of positive or neutral impact across multiple properties, including pharmaceutical properties, pharmacokinetic, ADME, and safety properties. That said, I do want to emphasize that although general trends and generic effects exist, the specific impact is always going to be context dependent. It's going to depend on the drug molecule, the target, the anti-targets, etc. And then finally, I'd like to make a pitch for Drug Hunter. Many property-based drug design strategies, tactics, challenges, and solutions can be found embedded in the articles we publish at Drug Hunter. So I encourage you, if you have a membership, read those articles. You're going to learn a lot. And if you don't have a membership, get a membership. And it's a great way to build your drug discovery knowledge. So finally, I'd like to thank the Drug Hunter team. These are the folks that are a real pleasure to work with on a daily basis. The research team is shown in lavender here on the left. I'd like to thank them for being great colleagues and helping me prepare this presentation today. But I'd also like to thank all of our operations and web development and design people for all the great work they do to bring our content to our membership. And finally, I'd like to thank all of the drug hunters whose work was included in this presentation. I thank you for listening, and I'm happy to take any questions.
Yeah, Lou, thank you for a phenomenal presentation. That was really, really very nice. Like many nice case studies and, very specific examples of how that necessary nitrogen impacts multiple different properties. So, very, very, very nice. And we got a lot of positive feedback from the audience already, and we got a lot of questions already. And I think we could start with this question here. So excellent, Yoshi says, excellent talk, Lew. I understand that you're mostly referring to aromatic CH nitrogen changes. Do you expect a similar necessary nitrogen advantage for non-aromatic CH to nitrogen exchanges? So, CH2 to oxygen exchanges are typically preferred by Yoshi, he says.
Yeah, one could imagine the outstanding oxygen atom as we increase the fraction sp3 of our molecules, and we have a lot more methylene groups in our molecules, aliphatic methylene groups. One could imagine that. So in my experience, yes, I have found that, for example, if you go from a cyclohexyl ring to a pyran ring, you can have profound improvements in a number of pharmacological and safety properties. People have written reviews on the hydroxyl group and even phenols. I would love to see a cyclic ether review. I don't think I've seen anything like that, but it's definitely something I think is both would be interesting, useful, and very timely, given how our molecules are starting to have higher fraction sp3s, more molecular complexity, and so forth.
Yeah. Oh, thank you, Lou. And yeah, then Gagan is wondering, and you already elaborated on that during the talk a little bit. If you could maybe elaborate a little bit more on the unnecessary nitrogen atom, you like brought already some disclaimers and some examples. Maybe if we could elaborate a little more in what cases like a nitrogen is somewhat unnecessary.
Yeah, so there have been a number of publications and reviews out there on this sort of matched molecular pair. So, you can look at my publications, but there's also publications from people at Pfizer, publications from people, I believe, at GSK. There's JMed Chem perspectives looking at this matched molecular pair. And you can get a sense from looking at those, you know, how to what how often you improve and how often you maybe worsen some of the pharmacological and safety parameters. It's not a panacea. There is no structure modification that's a panacea. Um, Again, it's all about probabilities. And if you have a lead compound with multiple aromatic CH groups, I would argue you absolutely should try this structure modification as one of the first things you try to optimize that molecule. I personally believe this structure modification is the most privileged structure modification in drug design.
Sounds good. We have another question here from Ragu. He says, as the complexity of our ring system increases, making CH nitrogen changes becomes even more complicated, especially in 6,6 or 6,5 ring systems. This change may not be possible with the nitrogen scan due to synthetic difficulties or time and resource lack. How do you or how do we, efficiently design the best one? And is there some predictive property that we could use effectively?
Yeah, so we have multiple pharmacological and safety properties that we're trying to optimize our molecules against. The easiest property to predict is binding affinity. So, free energy perturbation works pretty well for this structure modification if you have a structure of your therapeutic target. And there are other structure-based predictive models. You can use Metasite to predict metabolism. So use these predictive models, but I would just caution that, some are more reliable than others. And, it's got to be a judgment call for the chemistry team about, do we invest synthetic chemistry effort in this or not? I would argue this structure modification has demonstrated its utility so much, and that this structural motif, aromatic nitrogen atoms, are increasing in approved drug molecules so much. I think that the data argues that this is sort of a structure modification that does warrant synthetic chemistry investment. And this, again, I'll say this is why I'm so excited about this new area of synthetic chemistry, skeletal editing, because rather than requiring a de novo synthesis of all of the ASA analogs, you could take an advanced lead compound and do a late stage functionalization. And this is why I think this area of synthetic chemistry is one of, if not the most exciting, one of the most exciting areas of synthetic chemistry right now. And this is, I believe, is going to have a profound impact on medicinal chemistry and drug discovery. And I know there are some more skeletal editing papers that are going to be published very, very soon that involve this structure modification. So keep watching. I think there's going to be a steady flow of skeletal editing publications that hopefully will reduce the barrier to trying this structure modification on your advanced lead compounds.
Yeah, no, great example for how medicinal and synthetic chemistry are co-evolving, right? With the need of bringing more nitrogen atoms in, the synthetic chemistry develops and comes with a fantastic method. Also, a great way for academics to collaborate with industry. Another question here from Martin. Very insightful and well-delivered presentation. Any word on intramolecular hydrogen bonds to those jumping on nitrogen atoms?
Yeah, so in previous talks where I've talked about this structure modification in the context of multi-parameter optimization, I've included pharmacodynamic properties like binding affinity and potency and selectivity and functional activity. I did not do that in this talk because this talk is about property-based drug design, which is basically everything other than pharmacodynamic properties. So by all means, you can read previous papers that I've published in this area to get a sense of that. But absolutely, when it comes especially to two ASA compounds, this structure modification can have a profound impact on conformation through all of those intramolecular interactions that I talked about earlier, and then obviously intermolecular interactions for molecular recognition. Yeah, that just wasn't part of today's talk, but it's in the references that are shown here at the bottom of this slide. You can find a large number of examples of that.
Awesome, awesome. Maybe a question of my own, Lou. I'd love to pick your brain on like you have great experience with CNS drug discovery. Is there an impact on you? You talked a little bit about the impact on permeability, sometimes efflux. What are your thoughts on impact on brain penetrability, like the KP or KPUU? Is there a kind of significant impact? And are there maybe some examples out there where nitrogen maybe masked with where masking an NH was beneficial for brain penetration?
Yes, those are out there. So generally what you want to do, because in general, the structure modification is going to decrease your permeability and increase your efflux. But if you're strategic about it, or you think very cautiously about it, it depends on where you put that nitrogen atom. So let's say, for example, you have a hydrogen bond donor in your molecule, and you put this nitrogen atom in a position where it can form an intramolecular hydrogen bond. There are examples out there where you actually decrease efflux and increase permeability. I have personal experience with that as well. It's all about again, context is everything. So, if you have a very exposed aromatic nitrogen atom, it's more likely you're going to have a negative impact on permeability and efflux. But if it's sterically shielded, or if you have, say, a trifluoromethyl group next to it, like I showed in one of the examples, and you can shield and decrease the hydrogen bond accepting strength of that nitrogen atom, you can have either a positive or a less negative impact on KPUU and brain penetration in general.
Yeah, sounds good. And yeah, we're almost at time, Lou. So maybe one more question I want to ask. We have a lot more from the audience, so very popular talk. Maybe last question. What are your thoughts on exchanging a CH with the CF rather than a nitrogen? So basically putting a fluorine on there, and how does that compare to the necessary nitrogen atom?
Yeah, so I think it was Paul Leeson and Rob Young in a 2018 JMed Chem perspective talking about lipophilic efficiency. they described four privileged structure modifications. This was one. The hydrogen atom to fluorine substitution was another. Hydrogen atom to methyl substitution was another one, and then what was it? Fluorine, and then oxygen, hydroxyl group. So those are four privileged structure modifications. So yeah, I would say that structure modification is a close second to the necessary nitrogen atom, but I might be biased.
Sounds good. All right, I think those are great closing words. So again, Lew, thank you for a phenomenal presentation. Really, really enjoyed this. A lot of very useful case studies for the audience. Thank you to our audience members. Very engaged. We had a lot of questions, so we really appreciate that. Keep the questions coming. We love to hear from you. And then we'll see you at our next event, and that'll be next Thursday on December 12th. And we'll have Rebecca Paul here talking about the impact of AI in drug design, which will be a very, very interesting topic. So I encourage everybody to sign up for that. And yeah, we'll learn more about how AlphaFold 3 and other breakthrough AI tools are applied in drug design today. And with that, I wish everybody a phenomenal rest of the week. Thank you again, Lew, for being with us today and sharing this great topic with us. And see you, everybody.